fellow biology students, especially to our dear Professor Maribel B. Cruz. I am Julian C. Bandi. In this video, I'm going to discuss about single hit of the target theory. One of my groupmates, Denise Ann Fermin, already discussed about its effects. But first, to understand it better, we need to know about few basic things like cell death, estimation of survival or plating efficiency, and nature of cell killing
and cell survival. Estimation of survival fraction or plating, and the nature of cell killing. Cell survival curve. It describes relationship between radiation dose and the fraction of cells that survive that dose. This is mainly used to assess biological effectiveness of radiation. Cell death. Cell death can have different meanings. First is loss of a specific function, differentiated cells like nerve, muscle, and secretory cells. Second is loss of the ability to divide. Proliferating cells such as stem cells in hematopoietic system or intestinal epithelium. Third is loss of reproductive integrity or the reproductive death. Relevant dose. 100 gray destroys cell function in non-proliferating systems. For example, nerve and muscle cells. 2 gray mean lethal dose for loss of proliferative capacity for proliferating cells. Survival. Conversely, survival means retention of reproductive integrity. The capacity for sustained proliferation in cells that proliferate. So this is the proof of reproductive integrity. The capability of a single cell to grow into a large colony, visible to the naked eye. A surviving cell that has retained its reproductive integrity and is able to proliferate indefinitely is said to be clonogenic. Estimating survival. In order to determine the surviving fraction, we must know the plating efficiency. PE is the percentage of cells in control batch that grow into colonies. In other words, those cells that survive the plating process. Derivation of survival curves. Cells have been taken from stock culture and placed in seed dishes, as you can see on the right side. Then irradiated to 0 gray to 6 gray and allowed to grow into colonies for 1 to 2 weeks. Colonies have been counted for survival data, as shown on the right side. So this picture on the right side, the number of colonies counted, plating efficiency, surviving fraction. As you can see, there is test tube that has cell suspension, and the cell suspension will be transferred on the seed dishes. Then the number cells seeded will apply the X-ray dose, and then always will have a control batch to determine the PE or the percentage of the cells. Surviving fraction. Equal to the fraction of cells that plate successfully and survive irradiation without losing their reproductive integrity to grow into colonies. A cell survival curve is graph plotted between surviving fraction on y-axis and absorbed dose on x-axis. Meaning, surviving fraction is equals to colonies counted divided by cells seeded times 100 or the percentage of the cell. In this picture, the cell survival curve graph is plotted. Random nature of cell killing. as ionizations produced within cells by irradiation are distributed randomly. So consequently, cell death follows random probability statistics, the probability of survival decreasing geometrically with dose. Quantization of cell killing. A dose of radiation that introduces an average of one lethal event per cell leaves 37% still viable is called D sub-zero dose. Cell killing follows exponential relationship. A dose which reduces cell survival to 50% will, if repeated, reduce survival to 25% and similarly to 12.5% from a third exposure. This means surviving fraction never becomes zero. So this picture on the right side, this is a typical cell survival curve for cells irradiated. A straight line results when cell survival from a series of equal dose fractions is plotted on a logarithmic scale as a function of dose on linear scale. The slope of such a semi-logarithmic dose curve could be described by the D sub-zero, the dose to reduce survival to 37%. D sub-50, the dose to reduce survival to 50%. The D sub-10, the dose to reduce survival to 10%. D sub-0 usually lies between 1 to 2 gray. Next is the mammalian cell survival curve. Shoulder region shows accumulation of sublethal damage. The larger the shoulder region, the more dose will initially be needed to kill the same proportion of cells. Beyond the shoulder region, the D sub-zero dose or inverse of the slope of the curve indicates the relative radiosensitivity. The smaller the D sub-zero dose, the greater the radiosensitivity. The graph of the typical mammalian cell survival curve is shown on the right side. So let's dive into the single hit model of the target theory. Single target, single hit inactivation model. Single hit on a single target within the cell leads to cell death. This generates an exponential cell survival curve which appears as a straight line on a semi-logarithmic scale. As you can see on the right side, there is a graph. You can see there the survival curve of the cell. This model is useful for highly sensitive human tissues if high LET or linear energy transfer radiation is used. Mammalian cells usually have a shoulder on their cell survival curves, which is not seen in this model. The major problem with this model is that there are too many parameters, especially in multi-hit. To know more about multi-hit, check on my groupmates report, which is Alexander Merata. She discussed about the multi-hit of the target theory. So, this ends my report or my discussion. Thank you for watching!